Hello, and welcome to the video for the chapter entitled Optogenetics and Clarity. This brief video will serve as an introduction to modern optical techniques for manipulating, recording, and mapping neural circuits in the study of normal neurological function and in disease. The goal is primarily to provide a conceptual overview, illustrate major takeaways with key examples, and in particular, use videos to illustrate these techniques in action. For additional detail, please refer to the main text. In this presentation, we'll cover primary motivations behind optical and genetic techniques for studying neural circuits, which will pertain to each of the categories of techniques that we discuss in greater detail. We'll then discuss 1. Optogenetics, an optical method of stimulating neurons. 2. Calcium imaging, an optical method for recording from neurons. And 3. Tissue clearing, a method which enables whole-mount three-dimensional imaging and molecular labeling of intact tissues, such as an animal's brain. Finally, we'll discuss how these techniques, which have become ubiquitous in the basic neuroscience research community, are rapidly making their way into humans, and what clinical applications we might expect to see in the not-too-distant future. As suggested by the term optogenetics, there are two primary attributes to these techniques that provide unique utility when compared to other techniques, for example, electrophysiology: the optical aspect and the genetic aspect. Light is powerful because it has both high temporal resolution and high spatial resolution, which makes it possible to target cells that are, for example, right next to each other. It also comes in a spectrum of colors, so that one wavelength of light could do one thing, for example, stimulate, while another could do something completely different, for example, inhibit, all at the same time. Equally important is the concept of cell type specificity, which can be accomplished with genetic targeting. We've known for a long time that even within a particular piece of brain, there exists immense heterogeneity in terms of cell types. A simple example is the commingling of excitatory and inhibitory cells, which of course have very different properties and functions. But cells also vary in their connectivity, functionality, and other differentiating factors. And we now have developed ways to uniquely target many of these populations genetically. Now we'll discuss specific methods which take advantage of the strengths of optical and genetic targeting. The first of these is optogenetics, which is the use of light to control the membrane potential, and thereby excite or inhibit these genetically defined populations. The fundamental unit of optogenetics is an opsin, or light-gated ion channel. Opsins are a class of transmembrane proteins that open or close in response to different wavelengths of light, and thereby allow charged ions to pass through, resulting in a change in the membrane potential. They are, in fact, distantly related to the rhodopsins that are central to the signaling cascade for sensing light in the human eye, but most of the widely used opsins in neuroscience actually come from marine microbes. The canonical opsins used for mammalian neuroscience are channel rhodopsin, or CHR2, and halorhodopsin, NPHR. CHR2 is a blue light sensitive cation channel, meaning that brief flashes of blue light cause influx of positively charged ions and therefore depolarization, whereas NPHR is a chloride pump, which when illuminated by yellow light, pumps negatively charged chloride ions into the cell and thereby hyperpolarizes the cell, inhibiting firing. These are just two examples. A large catalog of opsins exist, both naturally discovered and designed by protein engineers in the lab, with a wide range of properties including light sensitivity, kinetics, the wavelengths of light that they respond to, and the ions that are conducted. So how does this work in practice? A typical optogenetic experiment would involve stereotactic injection of a virus encoding the opsin into the nucleus of interest, along with a fiber optic implant which allows for controlled illumination. At the time of the experiment, the fiber optic is connected to a light source, such as a laser, that is controlled by some function generator with defined stimulation parameters. Much like with electrical stimulation experiments, neural activity can then be induced or inhibited with the flip of a switch. For example, in one of the earliest demonstrations of optogenetics in action, neurons in primary motor cortex were stimulated, which in this mouse robustly drives circling movements. Since its initial invention about 15 years ago, optogenetics has been used to study many complex behaviors, such as learning, social behavior, or those that are involved in hunting, as shown here via stimulation of the central nucleus of the amygdala, which the authors of this study are then able to further deconstruct into the pursuit and actual biting process of hunting. Optogenetics has also been used to study fundamental neurological disease processes, for example, the role of neuronal activity in enhancing recovery from stroke or in driving growth of tumors. From applications in psychiatric disease to pain, epilepsy, and movement disorders, the list of both fundamental and disease processes is quite long and rapidly expanding. For more details and pointers to some high-quality review articles, please see the main text of this chapter. Now, the complement to writing in neural activity is, of course, the ability to read out the activity of the system. And we'll now discuss another optical technique that allows for the reading of neural activity from many individual neurons, called calcium imaging. The way that this works is that intracellular calcium concentration is known to be coupled to spiking activity of any individual neuron. So if there were a chemical sensor that could track intracellular calcium and convert that into a fluorescent signal in real time, that could serve as a reliable proxy for the underlying electrical activity of every neuron that you could see in a field of view. And indeed, the strategy turns out to work quite well, and in this situation, it might be easiest to just look at an example. So here, we have an example of where they've patch clamped a neuron in vitro and also expressed a calcium indicator. So you can directly see how the fluorescent activity corresponds to the electrophysiological changes that are occurring. And so as you can see, spikes correspond quite directly to increases in brightness. So to illustrate how this works at scale, you'll see here a large field of hundreds of individual neurons, and their brightness at any given time is a reflection of the number of action potentials being generated. And now, since you have a video feed of the neuronal activity, it's not too difficult to imagine tracking the brightness over time separately for each of these cells, and thereby having single cell level information as to the activity dynamics of the system of hundreds to thousands of cells simultaneously. Finally, let's touch on optical methods for mapping the three-dimensional anatomy of neurons. Connectivity of neurons is, of course, one of their most critical properties, but whereas a single neuron can project its axons across many centimeters in the brain or spinal cord, classical histology techniques force us to section tissues into micron-thick cross-sections, and so we lose all of that long-range information. One way is to try and create a 3D reconstruction based off of these slices, but as you can imagine, that's a hard problem. On the other hand, tissue clearing solves this problem by taking a large biological specimen and through a variety of chemical methods renders it transparent, as exemplified here by a mouse brain before and after clearing. This allows us then to visualize the entire brain in its intact state without the need for sectioning, thereby allowing simultaneous visualization of both large-scale macroscopic anatomical features as well as single-cell level high-resolution features. The most advanced of these techniques also preserves the molecular identity of this tissue, so they can still be stained or labeled with immunohistochemistry or in situ hybridization probes, which then provide even more detailed molecular information. For example, here we're labeling different types of cells, including parvalbumin-positive interneurons in red and GFAP-positive glial cells in blue in this mouse hippocampus. It's easy to imagine that the problem of three-dimensional histopathology has many applications, including accurately mapping the margins of a human tumor. In principle, as tissue clearing techniques mature, it may one day be possible to perform intraoperative pathology in 3 dimensions, alongside rich molecular profiling, which would provide significantly greater accuracy and confidence than looking at individual thin sections. Tissue clearing is performed entirely ex vivo, and therefore poses minimal clinical risk to patients, and development along these lines is well underway by numerous groups. Early studies have shown that this works really well in human specimens, both in fresh tissue as well as archived specimens, such as those stored in tissue banks. Turning our attention back to optogenetics and calcium imaging, as you can imagine, these of course have significant potential to one day be used as next-generation deep brain stimulators or brain-machine interfaces that offer single-cell resolution and cell-type specificity, either separately or in tandem in some closed-loop way. But to get there, there are a number of important safety issues that have to be rigorously tested, as well as many technical and practical challenges that must be addressed before such systems could work in the human brain. One of the primary questions surrounds the issue of gene therapy. After all, the central idea is to engineer human brains to express proteins from algae or jellyfish. To this end, numerous studies have been performed looking at feasibility and at long-term safety and performance of these protein systems in non-human primates, where the evidence so far is very promising. For example, this study evaluating long-term feasibility in a small cohort of squirrel monkeys in which channel rhodopsin was shown to reliably elicit time-locked spiking, and also there were no signs of abnormality on either imaging or lab values over 1 year following the virus injection. On the calcium imaging side, a proof of concept study in rhesus macaques recently showed that an optical brain-machine interface was able to reliably decode motor activity of a behaving monkey using only calcium activity recorded from the dendrites of the deep layer neurons in motor cortex. The safety of gene therapies in general is becoming more widely accepted, with numerous FDA-approved gene therapies for genetic diseases or hematological malignancies, and numerous ongoing clinical trials with direct virus injections occurring in the brain. There have also been a small number of phase 1 studies where opsins have been delivered into the eyes of patients with retinitis pigmentosa. As of right now, the results of these studies are still pending, but it's likely that numerous future directions in terms of clinical studies using these tools will begin in the near future. We believe it's an exciting time for optical methods in neuroscience, both in terms of their development into clinically useful diagnostics and therapeutics, and also for the broader impact that their use will have in elucidating new neural mechanisms and targets for treating disease. Thank you for watching this introductory video, and if you'd like any additional details or references, please do take a look at the main text of this chapter, and don't hesitate to reach out to any of the authors with any questions.